Now, bacterial toxin, remember just 2-3 things you have to remember here. The first thing is not all bacteria secrete the toxin, okay? Not all bacteria, you should rather write it down in your note that not all bacteria, okay, not all bacteria produce the toxin. Fine? Okay, there are some, okay, there are some bacteria which produce the toxin. Fine, no problem. And these toxins are, you can say, generally kind of disease causing agent. Okay, these toxins are basically a disease causing agent. Okay, it can be the protein, okay, which is you can say expressed by the bacterial gene, understood, or it can be the polysaccharide, it can be the lipopolysaccharide. Okay, so now we are going to discuss that in which you can say category of bacteria have the protein type of toxin, and in which case of the bacteria have the lipopolysaccharide kind of toxins. So for that division we have to classify the bacterial toxin. Okay, we have to classify the bacterial toxin. So this bacterial toxin, which is produced by the some bacteria, they are categorized broadly into the two categories. Okay, the one category is called as the exotoxin. Fine, and another group is called as the endotoxin. Exotoxin and endotoxin. As the name suggests, the exotoxin is present in the both. Which kind of both bacteria? Yeah, the gram. Both gram negative and gram positive. Both bacteria produce the exotoxin. If we talk about the endotoxin, it is only produced by the gram negative bacteria. Okay, so the first difference you should write it down. The exotoxin is produced by the both category of the bacteria. Yeah, the gram positive, okay, and gram negative. Okay, and if we talked about the endotoxin, it is specifically produced by the gram negative bacteria. Gram negative bacteria. The exotoxin's chemical nature is it is a protein. Okay, it is a protein, whereas the endotoxin's chemical nature it is a lipopolysaccharide. Polysaccharide. Clear? No problem. If we talked about their toxicity, who is more toxier? This exotoxin. Exotoxin is way more, you can say, potent, or it's way more toxic as compared to the endotoxins. There is one estimation which I am going to discuss with you, that 1 microgram of tetanus toxin, 1 microgram of tetanus toxin, okay, can kill 1 adult human. Write it down. It's worth to mention here that 1 microgram, okay, of tetanus toxin, okay, can kill adult human. Got the point? No problem. Now let's go a bit depth in understanding of the exotoxin and endotoxin. Okay. And our vaccines, all the vaccines we discovered, they are meant for basically dealing with this toxin. Those all antibiotics, okay, antibiotics, they are also, you can say, functional, operational at this toxin, for suppressing these toxins, killing the bacterial toxin, dealing with the bacterial toxin. So we will going to, you can say, the comparative study between the exotoxin and endotoxin. Okay, and another one is your endotoxin. The first thing you understood about the exotoxin is it is produced in the both category of bacteria, whereas if we talked about the endotoxin, it is only specifically produced in gram negative. Gram negative. Okay, another one you understood is if we talked about this bacteria, you can say the endotoxins chemical nature, it is a proteinaceous, fine, whereas if we talked about the endotoxin, it is a lipopolysaccharide. Relatively, it is a more toxic. Okay, and it is a comparatively it is a less toxic, but both are, you can say, toxic. Don't think that okay, it is a more toxic, which means we have to only take precaution from the exotoxins. No dear, you have to deal with also the endotoxin. Okay, it is the, if we talked about the exotoxins, it is not a heat stable. These toxins are not heat stable. You can say the moderate temperature, approx the 60 degrees Celsius, can inactivate this toxin, whereas these endotoxins, although it's a less potent or less toxic, but it is heat stable. It's a heat stable, means it needs the high amount of heat to be inactive. Okay, to be inactive. But you should write down some of the bacteria, some of the exotoxic bacteria, exotoxin produced bacteria, which will not inactivate by this moderate heating, and that the name is Staphylococcus enterotoxin. Staphylococcus enterotoxin. Staphylococcus enterotoxin. This toxin, okay, and one of the strain of E. coli, they will, you can say, not become inactive by this temperature. It need a high temperature. So it is a notable bacteria which become not, you can say, the inactive by this. Now if we talk about the endotoxins, this endotoxins is mainly, you can say, produced by the cell lysis. And bacterial cells is degraded, okay, and the bacterial cell is degraded, then it produces some of the endotoxin. Got the point? Yeah, no problem. Now the another things which you have to keep in your mind is that these endotoxins, they are involved in the septic shock. Okay, they are involved in the septic shock. Now what I mean by the septic shock, I mean by the septic shock is that it produces the fever. Okay, it produces the high, you can say, it's lower down the BP, and there will be a thrombus formation. Okay, so fever, okay, then thrombosis. Okay, fever, thrombosis, okay, and hypotension. Okay, along with it there is a shock. So these all symptoms when appear in the patients, we referred patient have the septic shock due to the endotoxin. If it is a bacterial disease, then it is due to the endotoxin. Got it point? Once again, we are discussing the bacterial toxins, okay, and we said not all bacteria produce the toxins, some of the bacteria produce the toxins, okay, and these toxins are mainly the two categories. Some are the exotoxins and some have the endotoxins. Now Exotoxins is a, you can say, present in the both gram negative and gram positive, whereas the endotoxin is specifically produced in the gram negative. This endotoxins chemical nature is a protein, and their chemical nature, the exotoxin chemical nature is, endotoxin chemical nature is a lipopolysaccharide. Now it is a more toxic. Okay, we said one estimation that 1 microgram of, you can say, the tetanus toxins can kill one adult human, whereas if comparatively the endotoxin is a less toxic. Okay. Now, these exotoxins, they will, you can say, deactivate in moderate temperature, the 60 degrees Celsius, and whereas the endotoxins, they are a heat stable kind of toxin. Okay, but the notable example of the exotoxins is the Staphylococcus enterotoxin and some of the strain of E. coli, which will not, you can say, deactivate on this moderate temperature. Fine. Now talk about the endotoxins. These endotoxins generally produce the septic shock, the fever, thrombosis, hypotension, and shock, collectively referred as a, you can say, the septic shock. Got the point? No problem. Now, once we understood about the exotoxins and endotoxin, let's move ahead and discuss one phenomenon called as the antigen switching. Which is one kind of protective mechanism of bacteria to deal with the immune system of host. Because you know we have bodyguards. Okay, and they are not, you can say, drunken bodyguards generally. Okay, they are alive bodyguards. Okay, and once they activate, it has started fight with the foreign particles. Okay, and make them to out from the environment, the host environment. Okay, so we are discussing one kind of process called as the antigen switching. This antigen switching is, first of all, one kind of protective mechanism, as we have in the form of inflammation. It is a one kind of protective mechanism for bacteria. Okay, it's a bacterial protective mechanism. Okay, it's a protective bacterial mechanism. What happened in antigen switching? You know, one of the successful characteristics of a bacteria is to avoid the host immune system. Okay, to neglect, or you can to escape from the host immune system, and this is done by the antigen switching. The bacteria, you can say, either alter the, you can say, their antigen, okay, or either they modify the antigen. The two possibilities is there in order to, yeah, write it down, in order to escape from host immune system. Bacteria either, you can say, switch on or switch off their genetic expression. Okay, either they switch on, okay, or they switch off the, you can say, genetic expression or genes. Okay, they mainly, you can switch on or switch off the genes which is meant for you can say producing the antigens, and this process is called as the, this process is called as the phase variation. Okay, write it down, this is called as the phase variation. Got the point? No problem. Now, and sometimes bacteria, in order to escape, in order to, you can say, invade the bacterial, the host immune system, okay, it modify the genes, okay, modification of genes, okay, modification of the genes, and this process is called as the antigenic variation. Antigenic variation. So what we understood by the bacterial switching? The bacterial switching is one kind of, you can say, cleverness of bacteria, okay, and this is meant for their protection. So in order to, you can say, escape the host immune system, in order to confuse the host immune system, okay, bacteria will undergo the two kind of, you can say, alteration or changes. Either those genes which are involved in, you can say, producing the antigen, expressing the antigens, they either switch off those genes, or either they switch on those genes, okay, to, you can say, confuse the immune system. And this process of switching on or switching off of the genes is called as the phase variation. So that's the one way to escape from the host immune system. The another one is it either modify the existing gene which is involved in the, you can say, the producing the antigens, they modify, and this type of variation is called as the antigenic variation. So now, concluding today's lectures, what we studied, we mainly discussed about the, you can say, the pathogenicity of all the microbes, okay, and we said those microbes which can cause a disease, we refer those microbes as a pathogen, okay, and the disease causing capability is called as the pathogenicity, and also sometimes alternatively used as a virulence. Clear? No problem. Then we said that there are patterns, okay, there are patterns, a specific pattern which is followed by the specific category of microbes. So in that we said we will discuss only the two. The one is the bacterial pathogenesis, the way bacteria produces the disease, causes signs and symptoms, and another one is the viral pathogenesis, the way the virus causes signs and symptoms. So we have we dealt with the detail, you can say, discussion over the bacterial pathogenesis. The first things we understood in the bacterial pathogenesis is that first bacteria have to enter in the host environment, okay, through the different, you can say, the portal of entry. Fine. Then we seen bacteria, in order to multiply, bacteria have to be stable on the host environment, okay. So because the host environment is highly dynamic environment, it won't allow the stability of the foreign particles, okay. So for that, bacteria undergo one process called as the adhesion, okay. Then we seen that not only adhesion, there are some other, you can say, the process to cause a disease, and one of the process is the invasion. A bacteria invade or penetrate into the mucosal lining, okay, by the help of some of the enzymes, okay. And then we also, you can say, the deal with the bacterial toxins, that some of the bacteria, not all, but some of the bacteria produces the toxins, okay, referred as a either endotoxins or exotoxins. We study the comparative, you can say, study between the endotoxin and exotoxin, and at last we seen one cleverness of the bacteria, that is referred as a antigen switching, by which it, you can say, escape the immune system, or it, you can say, invade the host immune system, okay. And in you can say, further lectures, next lecture we will deal with the viral pathogenesis. So thank you with having me. Bye. Have a nice day.